He earned his PhD in molecular and cellular biology at UC Berkeley, his AB in biology from Harvard. He's been basically an entrepreneur ever since he left academia. And share with you today what he's currently working on, as well as the kind of process of that journey. We've had the pleasure of working together at Cavalry Biosciences and Jupiter Bioventures, which I'll tell you a little bit more about today. And with that, Ned, take it away.
Can you guys hear me? Thank you, Zach. Thank you very much. And it's also it's wonderful to be here, and it's a super pleasure to work with Zach, who has been a delightful and ever-present collaborator over the last 4.5 years. So I'm going to talk today about a lot of hard things in biotech. And I want to begin with this. So this is a painting by Peter Bruegel, and he painted it in the middle of his career, a recent Renaissance painter. So this is about 1558. He called this painting a landscape with the fall of Icarus. So when I look at this painting, I actually don't see Icarus. I see farmers farming, I see shepherds shepherding, I see boats putting up their sails and going to sea. But then, if you look carefully, in the lower right-hand corner, you see Icarus, face down, ass up, drowning. Icarus is there because he tried to do something very hard and failed. He built some quasi-bioengineered biotech wings out of wax, tried to fly too close to the sun, and he died. Why am I starting this talk with such a bummer of a story? It's because almost everyone in this room is a biotech person, a biotech entrepreneur, trying to do hard things. And if you have not already experienced this, you will at some point experience being Icarus. You will try to do something very hard, and you will fail. And if you take anything from this talk today, what I hope you walk away with is that this is okay. It's okay to be Icarus and to fail. And I think it's important psychologically to be comfortable looking like an idiot, at least for a while. So what I want to do today is talk about the last 25 years that I've been building biotech companies ever since the last year of grad school. And what I hope to share are some of the principles that I was able to extract from this and I want to share with you. And then talk a little bit about how we're trying to give these principles life in a new company building vehicle called Jupiter that I'm working on with a bunch of people in this room, Zach included. So I'm going to cover 25 years in about 15 minutes. So that's a lot of temporal compression, so please excuse lack of detail. So I founded my first company while I was just finishing grad school in 1999. It was called Syrrx. I'll talk more about it, but it was a high-throughput structural biology company, meaning we solved hundreds and hundreds of crystal structures very quickly using robots, which was fun. Then I joined Versant Ventures as an entrepreneur in residence, and I founded two companies, Achaogen and Kythera. By the way, Syrrx got 2 approved medicines, Achaogen generated 1 approved medicine, and Kythera produced a 4th approved medicine. Now, then I joined Arch Venture Partners as an entrepreneur in residence, and doing that again, and founded Unity Biotechnology in about 2012. I regard this stuff as the past. Now we are in the present, and more recent activities we'll talk about today are Cavalry, that I co-founded with Jan van Derzen and Tim Stowe, who's out there. And then we'll talk at the end a little bit about Jupiter. And Jupiter is a new way to build companies that tries to take a bunch of the lessons of the last 25 years and use them to be hopefully more successful. So my biotech career began with this. So what is this? It's a peptide bond. And I was a junior in college, and George Whitesides, my organic chemistry professor, put this up, and he said, this is how nature uses chemistry to drive the biology and the biochemistry of life. And he said, it is the bond angle freedom and constraint of this chemistry that allows nature to build this. This is a protein, the three-dimensional micro machines that drive the biochemistry of life. And when I saw this, and I saw how basic chemistry could give rise to this beautiful thing, I said, I want to do this for the rest of my life. So this was all happening when this crystal structure was front and center in medicine. What is this? This is the structure of HIV proteins. This is the little machine that HIV that causes AIDS makes to cut its polyprotein up into smaller bits to form a lethal infection. And what you see in this teal and yellow are two HIV protease inhibitors, sequenavir and ritonavir, built on the back of this structure. And it was because of these structures that these drugs were able to be built so efficiently. And it was, in a sense, these structures that converted AIDS, a death sentence for a generation of people, into something today that is a managed condition in the developed world. And the issue, when I sort of realized this, was that crystal structure making is hard, or at least it was hard. And I thought, myself and my PhD advisor, Ray Stevens, we asked ourselves, could we take the principles used from automating the car industry and mix that together with modern molecular biology techniques so that we could make crystal structure solving really fast and cheap? And we founded a company to do this called Syrrx, and we built a bunch of really cool robots, which were essentially cast-offs from the automotive industry. And we hired a bunch of guys who worked at a company called Saturn Cars, which is, if you know what that is, you're old as I am. But they built the drivetrain from the Saturn L class. And they had never done biology before, but they were down for it. And so we got together and said, we want to make robots that will automate growing bacteria, purifying tagged proteins out of bacteria, setting up as many as 1 million crystallization experiments in a single day, keeping those crystallization experiments crystallizing, and then finding the crystals using very early neural networks so that human beings didn't have to look at 1 million drops per day. They could look at, say, 200 and find the crystals. At our peak of operations, we were doing 23 times the number of experiments based on our best estimate of every crystallographer in the world in that era working together could do. It was a massive experimental throughput enabled by all of this automation and software. We did something else cool. You guys know what this is. It's the advanced light source. It's a synchrotron, which basically means you zip particles around in a ring close to the speed of light, and they emit high energy X-rays. And so we were the first company to do something a little unorthodox. We built a little hutch inside that where we could collect the high energy X-rays, blast them through the crystals to get high resolution pictures of the shapes of proteins. And this was an unusual decision, but it made us go really fast. This is 1 month's output we are running at peak. These are a bunch of essential bacterial gene products, and someone said, you know, it would be really valuable if you solved the crystal structures of lots of these sorts of proteins. Well, that was a stupid idea. It turns out no one cared. And we spent the 140 million or so we raised building all of those robots, solving lots of structures that no one really cared about, before we got down to about $10 million and asked ourselves, why are we doing this? And the company was in a whole bunch of distress, and our CSO, who was very wise, said, you know what, no one cares about lots of structures. What they care about is a few structures that really matter. And so we focused our energies on a handful, including this one, which is the structure of DPP4. We were the first pharmaceutical company to solve this structure. And it allowed us to design this molecule here. And back in the day, this was called Syrrx 322. Later, when we got the medicine approved, it was called allogliptin. And it's an inhibitor of this enzyme that's involved in processing, of all things, GLP-1. It's a GLP-1 inactivator. And so when you put this little molecule in there and you gum up the works, you get more GLP-1. Now today, of course, we have GLP-1s being all the rage, but I guess we were there before it was cool. And not only did we get this molecule approved, we got its sister molecule approved as well that has a much longer half-life. So at the end of, right as we were getting this first molecule into people, we were running out of money, we were saved because a Japanese company, Takeda, came and bought us and took these molecules all the way through clinical development, all the way to approval. But it was kind of a bummer to do all of this and build all this cool technology and then find yourself having done this thing essentially without any strategy. But we were all young, and what principle did I sort of take from this? Well, I think the principle that I mostly took is that when people give you too much money, when you have capital excess, you wind up essentially dying because decision-making quality deteriorates dramatically. The best decisions are made when you are under capital constraint. At the end of that company's capital, when we finally made the decision to go after structures that people cared about, that's when we were really in the biotech business. Before that, we were just playing. Now, the sort of corollary to this statement is that capital constraint, when you have just enough money to do what you need to do, that is life. That is when all of the best decisions wind up getting made. So this is a tough principle to incorporate when someone hands you too much money. Because even if you know this to be true, the human brain doesn't like to be constrained when there is excess around. So you have to be very knowledgeable about the psychology of this state as you build your companies and you finance them. My next company began when my boss at Versant, a wonderful investor named Cami Samuels, who's invested in all my companies over the last 25 years, she said, you've become quite a small molecule nerd. And you need to shake it up a bit. I'm sending you to a medical meeting. And I didn't want to do this. And she said, in fact, I'm sending you to the Academy of Dermatology meeting. And I sort of groaned, and I flew there, and I was walking around, and it's all these symposia about taking fat out of people's asses and putting it in their face. And I thought, has it really come to this? This is my life now. And I'm wandering around all dejected, and there was this seminar that said the molecular biology of the aging face. And so I thought, molecular biology, I'm saved. I'm going into this room. And I walk in, and the professor then puts up this slide, and it shows the EGF signaling pathway. And he said, this is why you photoage when you go in the sun. That this signaling cascade gets activated, and it drives the production of commercial proteases that chew up your dermis. And I walked up to him afterwards, and I was aware of a series of drugs that acted on this biology, and I said, have you ever thought of making a cream out of Tarceva? And he looks at me, and he said, what's Tarceva? And that was the moment I realized that the people who were doing this kind of weird backwater biology of aesthetics had no idea of the vast pharmaceutical chemical armamentarium that could be repurposed, taking from medicines for legitimate diseases and pointing them at something that is not a disease, but is human appearance. And so we founded this company, Kythera, and we took 4 molecules into the clinic. 3 failed for different reasons, but this one worked, and for those of you that are sort of cholesterol biosynthesis nerds, you might recognize what this is. This is a bile acid. It's a detergent made in your GI tract to solubilize fatty acid, or essentially fats that you eat in your diet. Now, guess what happens when you inject that into fat tissue? Something like this. It explodes violently within seconds of exposure. And so we advanced this molecule to destroy, of all things, the scourge of the double chin. And this is a patient from one of our phase 3 studies. This is an actual patient prior to treatment with this molecule. And this is after. So let's do that again. Before, after. So it's a pretty profound result. And so we took this company public, and this was fun. I had never gotten to do this before. And we then eventually got an approved medicine. It was about a decade, and we actually commercially launched it ourselves. And then our competitor, Allergan, bought us for about $2.1 billion. So for this company, we raised about 110 million privately, and then about another 350 million publicly. But what's cool about this is that our Series A investors that included Versant and Arch and ourselves as founders, we made 100 times our money between that Series A round and when we sold the company. And that's a pretty extraordinary result. And we did that because we figured out for the first $1 million that this molecule, that bile acid, blew up fat cells. So the future had taken root in our present. And for the next 10 years, the trajectory of the company was simply following that logical path. And so what principle do I take from this? Well, it's that tackle your biggest risk first with your very first money. Take that risk that is in your future and put it in your rearview mirror inexpensively, because everything then flows naturally after that. So my next company begins, as we all begin, as a single cell fertilized by mom and dad. And over the arc of your life, you will divide a bunch of times. And most cells in your body can divide about 50 times, at which point they will pull an emergency brake and stop dividing forever. And cells that do this, we call them senescent cells. Now, there's a bunch of other stresses that can induce this state. And no one had known whether these cells that accumulate in you, and when you're little, like my son here, when he was 8, had very few of these, and my stepfather, who died at 87 from Alzheimer's, had a lot of these. No one knew if these cells were good for you, bad for you, or neutral. And so my co-founders, Jan and Judy, did an experiment where they asked what would happen if you were to delete these cells from a creature during its life. Well, this is what happens. So these mice are litter mates, born from the same mom. They are essentially genetic clones of one another. The one on the left, and these guys are about 70 years old, if in human years. So, the one on the left is blind, osteoporotic, and frail, has kidney dysfunction, cardiac dysfunction, it has obvious cataracts. Its litter mate on the right has many of these features, but they are diminished. And it also lives 35% longer on average. So this is a cool result. So we thought, let's form a company based on this. Oh, before we founded the company, it was also clear from the literature that these cells were implicated in a bunch of biology. These cells were driving kidney dysfunction, they were causing lung fibrosis, they were causing arthritis in your joints. And so we created a company called Unity to pursue this biology. And it was a really fun company to work at for the first 5 years. So we had 8 high-impact papers. We had Breakthrough of the Year 1 year from Science. We had 120 employees. Over the arc of the company to date, it's raised $500 million. And we decided to tackle osteoarthritis first. So for those of you that are too young to have experienced osteoarthritis yourself, it is the primary reason it hurts to be old. So allow that to wash over you. If you could get rid of this disease, imagine being able to be old and having some of the other problems of being old, but not having your joints hurt. It would profoundly alter the last third of the human life experience. And so what we found at Unity was that the synovial membranes of patients had elevated amounts of senescent cells. These are the membranes that surround your joints. And we hypothesized that eliminating these cells, which we thought were driving the chronic sterile inflammation associated with OA, could cause that disease to go away. And so with that as the promise, we got to take this company public, so we got to do this a second time. And then things started to get real. we did our first study. And we did a phase 1 study, which are often fraught and often give falsely positive views of reality. But in this first study, we saw a signal where if we put this drug that we thought was removing senescent cells from people's knees, we thought we saw a change where pain went down. We proceeded forward and did a phase 2 study. And when we did a properly controlled, properly randomized phase 2 study, that mirage of a clinical signal we saw in phase 1 went away. And in retrospect, when we went back and looked at the phase 1 study and the way we did it, the phase 2 study was telling the truth, and the phase 1 study was not. When this happened, Wall Street, perhaps correctly, decided that this idea was not a good one, and the company went from having $1 billion market cap at the peak down to a market cap that is 50 times smaller today. And our head count went from 120 people down to about 12. So this was awful and a tremendous loss of promise and pride and team. And the company was forced to do some deep soul searching. But we retrenched and we began looking at biology in the retina, specifically the vasculature that feeds the retina, the arteries and veins behind the retina. So what are we looking at here? This is a mouse retina under stress, and the reason it looks like a four-leaf clover is you cut it out and you flatten out it. So what you see is in the center of this retina, the vasculature is all dead. This is a mouse that's experiencing oxidative stress in a model that essentially copies diabetes. And as you know, one of the complications of diabetes is blindness. And what you see at the periphery is all of this diseased vessel growth. What happens in this mouse when you eliminate senescent cells during the stress? You get a retina that looks like this. The center is alive, the diseased tufting at the edges is dramatically reduced, and this data was good enough that we decided to try again. We went into people with diabetic eye disease, and I'm going to show you some human data. This is a picture of a human retina from the side. This is a normal one, and look at its beautiful architecture, it's very organized. Now I'm going to show you a retina from a diabetic eye. As you can see, it's all distorted geometrically, it's full of fluid. This is what a diabetic eye looks like. This is why diabetes can drive ultimately blindness. So this is a patient that received a different molecule than the one we took into the knee. This is one specifically built to destroy senescent cells that live in the vasculature. This is a patient before injection. This is a week after, 4 weeks after, 8 weeks after. A remarkable change in a human disease. Now, it's not just these pictures of retinas that get better. Patients gain vision. 6 letters of vision on an eye chart at the DMV. Now, 6 letters for an older person is the difference between being able to drive and not being able to drive. Now, we shall not pause at this juncture to discuss the wisdom of old people driving. And my mother's here in the front, so I'm sure she's going to give me an earful from this later. But the point is that this can produce a profound effect in humans. And as far as we know, this is the first evidence that removing senescent cells, a process we call senolysis, can actually take a human disease and send it backwards. And 1 out of 3 patients that get this medicine, a year after they were given a single dose of this medication, their disease has not returned. Meaning eliminating these cells sends that disease backwards without having to keep redosing this medicine. So we remain optimistic, but the company has been permanently damaged through its history. So what principle can we all take from that? Well, I think it's pretty obvious. No one in this room, please don't you dare go public until you are really sure you're ready. And we can talk later about what does ready mean But clearly, Unity was not ready for that. So what are the principles? Capital constraint is life. Tackle your biggest risk with your first capital. And finally, please, I know it's fun, but don't go public until you're ready. So now I'm going to shift gears and talk about the present. I'm going to talk about Jupiter, which is a new company creation model that we have created in collaboration with Mission Bio as well as Mayo Clinic. And the core idea behind Jupiter relates to this. This is a black box. The big challenge in doing biology is that biology is a black box. When you start a biotech project, you do not know at the beginning if it is going to work out. Yet, as founders, we are asked to go to our investors and go to our teams and say something like, I swear I have enough evidence, I guarantee you this is all going to turn out fine. And you have to make these statements to raise the money, to organize your team, and you're not allowed to peer inside that box until years and tens of millions of dollars later. But does it have to be that way? What if there was another way to do this? What if instead you went out to the world of ideas and you evaluated a bunch of these ideas, say a few dozen, and you had some criteria of merit, and say 8 of these looked interesting enough to tackle. This is kind of what we do at Jupiter. And we will then spend a few $1 million, up to about $2.5 million, to try to see whether or not, when you peer inside the black box, it looks good or if it looks bad. Now, half of everything when you do this dies right in infancy, as it should, because the project did not merit proceeding forward. At which point we'll spend a little bit more money on these ones where we have some conviction. More die. And eventually you wind up with just a few projects left that at Jupiter we'll commit up to $20 million to. And we call these projects moons. And right now at Jupiter we have 4 moons that are up and going. And this model of incrementally removing risk with small amounts of capital allows you to honor the first principle. We maintain capital constraint. We also force ourselves to decide what is that biggest risk and spend those early dollars solely on that biggest risk. We also try to build molecules that either are clinic ready or are the great-grandparents of molecules that can become clinic ready. And what this does is it sets these companies up with those principles from their earliest moments of creation. We also have 3 moons that are sort of in the wings in states of evaluation, but before we spend any money. And I would loathe to ignore these, the deorbited moons. So these are the ones that we've already killed. So we've killed 3 moons so far for about $2.5 million each. We did our very best. We identified key risks, but they did not survive. And this model only works if you ruthlessly kill bad projects. And anyone who's a kind of comics fan might recognize that that is Superman's homeworld Krypton exploding, so it's not actually a moon. So I'm going to talk about one of our first moons that we're working on, Cavalry. This is one that I co-founded with Tim Stowe. And the premise behind this moon is pretty simple. What if you wanted to send a growth factor anywhere in the body? What would you do? Well, there's lots of things you could do. You could send a growth factor to rebuild peripheral nerves. You could have it regrow glands that have regressed with aging. Or you could send it to muscles, which is the one piece of data I'll talk about because it's pretty cool. We have programs in all those things I described. Now, in muscle, we built a molecule that kind of looks like a off-kilter antibody. So what you'll notice is it's got a part that sticks to the muscle fiber. It's got a part that's got a growth factor, IGF-1, fused to it, Then it's got a part that looks like a classical antibody to give it antibody-like pharmacokinetics. When you inject this in an animal, it decorates the surface of muscle fibers, and it does so almost exclusively. And this project is the brainchild of Britt, who is floating around out there somewhere. This took years to find the target, and then additional time to engineer this molecule. But look what happens when you take Britt's creation and you put it into a mouse for 2 weeks. You get a yoked mouse. All right, so this is a mouse on vehicle. This is a mouse with 4 doses of this molecule. And it is an extraordinary phenotype, to the point where any scientist who's working on this experiment, you can't really do this blinded because the cage with the drugged animals gets too heavy. So we're applying this in a variety of places. I'll show you some pretty cool data from a severe muscular dystrophy. So this disease is called FSHD. It's a bit of a mouthful, but it's facioscapulohumeral muscular dystrophy. It is the disease most widely known because the founder of Lululemon suffers from this disease. It is something that will gradually cause loss of your muscles in your back and your hands, and it's a tragic disease. It's also the most prevalent muscular dystrophy in the world, and the disease happens because of mutations in your genome that give rise to expression at the wrong time of this transcription factor called DUX4. And it's a transcription factor that tells your muscles cells to die. And you're not supposed to make this for most of your life. You're supposed to make it during development. But when you do, the muscles die inappropriately. So when we inject our molecule into mice with this disease, it largely preserves muscle weight, but most importantly, it entirely reverses the loss of muscle strength. And our collaborators, who have been working with all of the pharma companies running, this is the foundation, the FSHD Foundation, that the Lululemon guys run, they've never seen a molecule fully correct the strength defect of this disease. So we're very excited about this. We hope to be able to create a medicine that can address this horrible disease. I'm going to show you one more moon before I finish up. This one is called Persistence, totally different universe than Cavalry. And to understand persistence, it helps to think about how do we, as we just sit here in our chairs eating pizza, fight cancer on a day-to-day basis before we have the disease. Well, what happens is cancers form in your body, probably frequently, though it's not clear how frequently they do, but by the time you're 18, most people have antibodies against melanoma, which is pretty interesting, which implies that your immune system is doing its job. So when you get a cancer and you're not actually manifesting with full-blown disease, your immune system has things called T cells. And these cells are essentially killers. And they swim in, they attack the cancer, and under normal circumstances, the tumor gets eradicated, and you just keep living your life. So it turns out one of the most potent ways to treat a human with full-blown cancer is to borrow this system using a technology that's been around for about a decade called CAR-T, and CAR stands for chimeric antigen receptor. It's where we engineer a specific biotech-built sequence of a receptor, and we put it into your T cells to turn them into hunting, killing machines. It's actually, in principle, it's a very simple approach. You take T cells from somebody's blood, you genetically modify them in a lab by putting in this special receptor that converts them into kind of a terminator, and you put them back. And you let your own immune system, modified and armed, fight your cancer. Now, this approach is more powerful than any cancer therapy for certain lethal lymphomas. So take acute lymphoblastic leukemia. It is 1.6 times better in terms of producing overall survival in patients compared to the most aggressive standard of care. For various different aggressive B-cell lymphomas, it's 4 times more effective at producing what is called a complete response. A complete response stands for if you scan somebody, there is no evidence of disease. So 4 times better than aggressive chemotherapy. Mantle cell lymphoma, one of the worst diseases you can get. It is 9 times better. And in fact, if you get mantle cell, which I really hope you do not, you have to initially I'll talk a little about this, but you have to undergo chemical chemotherapy. You have to undergo immune checkpoint inhibitors and BTK inhibitors before you get to this massively powerful medicine that is 9 times better. And then for multiple myeloma, it's 6 times better. So this sounds marvelous. This is a powerful tool that should be used everywhere. However, CAR T technology is expensive, it is slow, and it is complicated. It costs $150 million to build the facility in which you do this. It costs $10 million to operate these facilities per year. It takes a month from the time a patient gets enrolled, when you get their blood and you genetically modify it and put it back in. Many patients will die because it's a last line therapy. They'll die between the time they get enrolled and the time they get the medicine. And it costs $1 million between the cost of the drug and the hospital utilization costs to do this therapy. What all that means is that these medications are typically 4th line. You have to fail everything before you get treated with this. Does that seem wrong? So does it have to be this way? What if there was a way to take this wildly powerful technology and turn it into a 1st line bedside therapy without giving up the power of autologous CAR T therapy? So we have an idea. So it works like this. You take blood from your arm and you get what are called your peripheral blood mononucleocytes. So these are just cells after you spin down the blood. 70% of those are T cells. And you take those cells and you squirt them onto this little sponge, which is about the size of a silver dollar, along with the virus that contains the receptor. Right now, as you're watching that stuff wake through, you are forming CAR T cells. Right now. And by 20 minutes, it's all over. You've formed all these CAR T cells in a process that we call, appropriately, spongeduction. So then what you do is you just take that thing with all of its CAR T cells and you implant it in your armpit. And it sits there spitting out CAR T cells for months on end. There's no GMP facility, there's no month-long growth. It is something that should be about 8 times cheaper in terms of the drug costs, and 14 times faster, because excluding the lymphodepletion that you have to get when you get these CAR Ts, this is a 1-day vein-to-vein procedure. So this is pretty cool, we're excited about it, and we're advancing this one forward as well. So, thanks for listening to all of this stuff. In closing, I want to leave you with a thought. So despite all of the sort of principles of don't do this and do that, I think it is better and nobler to act, to be biased toward action. What that means is it's better to throw yourself, body and mind, completely into projects that you find magnificent and that can make your soul sing. It is better to do that than to simply analyze the hell out of things and ultimately do nothing. Now, my ancient Persian isn't very good, so I'm going to try to paraphrase Syrrx here, but achieving great things requires embracing great dangers. And it's also way more fun to do it that way. So thank you all.